Hello, AP Bio folks. Today we are going into the last part of protein synthesis. We are going to talk about the process of translation. We'll go through a little bit of notes and we will continue our modeling activity. So today you will need the messenger RNA that we made during transcription. You will also need the large and small subunits of your ribosome that you made on Friday, and about 15 transfer RNAs that you should have also cut out on Friday. You will also need some tape and a scissors. So grab your materials, and then we will get started with some translation. So translation is the process of using the code in the messenger RNA to actually produce a polypeptide chain. Just like transcription, we will talk about kind of the three steps: the initiation or the starting of this process, the elongation of that polypeptide chain, and the termination, the ending of the process. Initiation of translation starts with the small subunit of the ribosome, which already has the first, what they call the initiator transfer RNA already attached. The small subunit attaches to the messenger RNA at the 5' cap. Remember, one of the purposes of that 5' cap was to help the ribosome get itself oriented in the right spot and moving in the right direction. So it attaches to that 5' cap. And then it begins scanning downstream along the messenger RNA until it finds the start codon, AUG. So that initiator tRNA is the one that is complementary to the codon AUG and carries the corresponding amino acid, which is methionine. Once it finds that start codon, that sets it in the right spot. It establishes what's called the reading frame. So the groups of 3 nucleotides from that point on will be correct during the reading of that message. Then the large subunit of the ribosome attaches, which creates what's called the translation initiation complex. There are other proteins that are involved in this called initiation factor proteins, and it does require some energy from a molecule called GTP to form. We're not going to worry too much about those pieces in our modeling, but just so you know, there are other proteins that are involved in this part as well. So we are going to take a minute here and model initiation of this process. So the first thing is you are actually going to need to write on your transfer RNAs as we go through the modeling part today. So on the first transfer RNA that you have, you are going to write in the nucleotide bases UAC. So you can grab your first transfer RNA right now and write in the bases UAC. And across the top, you're going to write the name of the amino acid. The amino acid that comes with this transfer RNA is called methionine. Then you are going to assemble your translation initiation complex. Both parts of the ribosome, the messenger RNA, and the initiator transfer RNA, which is in the P site of the ribosome. This is where you'll need to take your third photograph for this assignment. Make sure to include your little identifier, whatever you chose, within your pictures. So this is kind of what this is going to look like. Let me move my extra materials out of the way for a moment. So you are going to take one of your transfer RNAs and actually fill it in, write the bases UAC across the bottom, write the name of the amino acid methionine in the top. Now remember, the way that this starts is that the small subunit of the ribosome starts at the 5' end. So here's my 5' cap that we added last week. And it already has this UAC transfer RNA attached. And so this is going to kind of scan down along the messenger RNA until it encounters that start codon. Here it is. AUG are the bases that are complementary to that UAC. So that tells the ribosome where to start. At that point, the large ribosomal subunit moves into place. I kind of try and get mine behind there with that transfer RNA in the P site. So that's how we initiate translation and start in the correct spot. Don't forget, this is where you have to take a photograph, throw in your little personal identifier so that I know your photo is yours. As we continue the process of translation, we are going to be reading the messenger RNA codons. We read those nucleotide bases in sets of 3. So genetic information is said to be encoded in a triplet code, which you can see in the codon wheel here. You have a version of this written as a chart. This one you may have seen in 10th grade. We kind of read this from the inside out. It is important to remember that codons are the triplets found on the messenger RNA. So the letters you see in these circles are the letters from the messenger RNA. So if we look for our start codon here, we have our AUG, which gives us the amino acid methionine right here. A couple of really important things to notice about this triplet code. There is redundancy in the triplet code. What that means, redundant, means things repeat. So there is redundancy in the genetic code. There are multiple codons. If we look here, UCU gives us the amino acid serine, but so does the codon UCC and UCA and UCG. That's redundant. There are multiple codons that end up giving us the same amino acid. You'll notice a lot of that redundancy happens in the third letter of the triplet. Sometimes that's called wobble in the genetic code. The third base can wobble or change a little bit, and it doesn't always change the amino acid. But even though there is this redundancy, several codons can code for the same amino acid, there's no ambiguity. There's never any question what a codon means. Each codon has one specific meaning. The codon GCG is always going to mean alanine, and it can only mean alanine. So there's no ambiguity, there's no question. The code is very specific, even though there is a little bit of repetition in there. Like I said, you have a version of this that is a chart that we will be using when we get to translating your messenger RNA in a little bit. So the next part of translation is elongation. We've gotten everything in the right spot. The ribosome is in the correct place. We have the first transfer RNA in there with its amino acid. Now we need to start making our polypeptide chain longer and elongating that strand. So amino acids are added one by one. There are elongation factors. Again, lots more proteins are involved in this process than what we're talking about. The ribosome reads from 5' to 3' along the messenger RNA, and transfer RNAs move in and out of those different sites on the ribosome. Remember, the A site can stand for amino acid, or you can think of as arrival. So the new tRNA moves into the A site of a ribosome. It then forms a chemical bond, a polypeptide bond between those amino acids. That bond, the formation of that bond, is actually catalyzed by the ribosomal RNA, which then removes that polypeptide from the transfer RNA in the P site, and it's now attached to the one in the A site. Then this process called translocation happens. The ribosome shifts one codon down, and the first transfer RNA is now finished with its job. It's in the E site, and it exits. I want you to kind of see this process in action, so I'm going to show you an animation real quick here, mainly so you can see how this process works.
Translation is the synthesis of a protein from an mRNA template. This process involves several key molecules, including mRNA, the small and large subunits of the ribosome, tRNA, and finally, the release factor. The process is broken into 3 stages: initiation, elongation, and termination. Let's see the process in action. Eukaryotic mRNA, the substrate for translation, has a unique 3' end called the poly A tail. mRNA also contains codons that will encode for specific amino acids. A methylated cap is found at the 5' end. Translation initiation begins when the small subunit of the ribosome attaches to the cap and moves to the translation initiation site. tRNA is another key molecule. It contains an anticodon that is complementary to the mRNA codon to which it binds. The first mRNA codon is typically AUG. Attached to the end of the tRNA is the corresponding amino acid. Methionine corresponds to the AUG codon. The large subunit of the ribosome now binds to create the peptidyl, or P site, and the aminoacyl, or A site.
This animation, just so you know, does not label the E site, but the E site would be over here to the left of that transfer RNA that you can see right now. This right here is the translation initiation complex. And now they're going to start showing how elongation occurs.
The first tRNA occupies the P site. The second tRNA enters the A site and is complementary to the second mRNA codon. The methionine is then transferred to the A site amino acid. The first tRNA exits.
So right there you can see the ribosome shifted over, and this is now in the E site. That first transfer RNA can exit because it has dropped off its amino acid.
The ribosome moves along the mRNA, and the next tRNA enters. These are the basic steps of elongation. As elongation continues, the growing peptide is continually transferred to the A site tRNA, the ribosome moves along the mRNA, and new tRNAs enter. When a stop codon is encountered in the A site, a release factor enters the A site and translation is terminated. When termination is reached, the ribosome dissociates and the newly formed protein is released.
So that just kind of hopefully put together these pieces a little bit for you so you can see sometimes this makes more sense when you see it happening in action. And as you saw in that video, termination happens, the ending happens when we reach a stop codon. And you'll see in your codon chart, there are 3 specific codons that don't code for amino acids. They just tell the ribosome that it has reached the end of that strand. A release factor protein comes in, it binds to the stop codon, and it releases that polypeptide chain, and it leaves through the large ribosomal subunit. And then all of the components of this process are reused. The small and large ribosomal subunits separate and go find another messenger RNA. The transfer RNAs all float off into the cell to get new amino acids and help to build other polypeptide chains. So now it's our turn to do some translation. I'm going to show you kind of what this part looks like, but as we work through this, just a couple of reminders, and then I will demonstrate this part for you. We are finishing up this modeling activity today. So you're going to do your translation, take your last photograph, and answer all the questions on the Google Doc that goes with this activity. This entire activity is due by Tuesday night. So finish it up and turn that in by Tuesday night. During Wednesday's live session this week, we are going to be doing a partner assessment. If you are not going to be able to make the live session Wednesday, please email me right away so that I can plan accordingly. And of course, you have Mastering Biology homework to continue working on. It's not due until Thursday of this week. So let's come back now to our translation. so that we can kind of see how this process works and how we're going to model this. So for each transfer RNA that you need, you will be filling out the bases and the amino acid using the codon chart that you got with your packet. So if I look here in my A site, my next codon is ACU. So I know the complementary bases on my transfer RNA, which I'm going to fill out over here, are going to be UGA. So you write in those bases. And then you have a copy of the codon chart that looks like this. So you can look up, the first base is U, second one is G, U, G, A. Nope, sorry, I am looking at the wrong bases. Remember, these codons are the messenger RNA codons. So you need to look at the letters on the mRNA. We are not stopping already. So ACU is the codon I am currently looking for. Sorry. ACU codes for this amino acid THR. If you flip your codon chart to the back, it will tell you what that amino acid is. So THR is threonine. So I'm going to write that name at the top of my transfer RNA. So I have now filled in my next transfer RNA with all of that information. And so now I can put that will move into my A site right here. Remember, that's the first thing that happens. The new amino acid moves into the A site. And then a peptide bond forms between the amino acids. As you do this, we're only taping together the amino acids, because then as the ribosome moves down like that, my methionine is now in the E site, which means that transfer RNA is going to exit. So what actually is going to happen here, I just need to pick this up for a second. I am actually going to cut apart my amino acid right here, because this one is sitting right here. AUG, sorry, I'm losing my messenger RNA right there. So AUG is in that one. This moved over to here. Here we are. So this transfer RNA in the E site now exits and leaves to go get another amino acid, and I am left with this chain of amino acids growing. Now in my A site, I have CGA. So once again, I'm just going to demonstrate one more for you. CGA is a codon that codes for the amino acid ARG. So I'm going to go look at the back here. ARG is arginine. And so I am going to fill out my amino acid. The amino acid is arginine. The anticodon here is going to be GCU. So I'm going to fill that in. That is going to bond in the A site right here. I am going to have a peptide bond, otherwise known as a piece of tape, that connects my two amino acids. And then these things will move. My ribosome will move down. It's actually the ribosome that moves. I'm just going to do the sliding this way because it's easier. And then the previous transfer RNA over here is going to be separated and leave the cell. So you can see here we have this growing chain of amino acids that were coded for by these codons. And you are just going to continue that process moving down your messenger RNA until you encounter a stop codon that will tell you to finish. Finish translating your messenger RNA, finish up this modeling activity and get that turned in. Let me know if you have any questions, and I hope you have a great rest of your day.